Welcome everyone to the home stretch. This is the last session of the day. This is part 2 of advances in measurements of atmospheric pollution. My name is Winston Luke. I'm the outgoing NOS secretary and want to acknowledge my co-chair, Brett Chicatel. So we have 6 talks coming up. We'll finish up a little after 5. So first off is Dane Blanchard from Trent University.
All right, thank you. So I'm Dane. I am a PhD graduate, or PhD student from Trent University in Peterborough, Ontario, Canada. And my thesis focuses on the evaluation of chromophoric dissolved organic matter throughout the Athabasca oil sands region in Alberta, Canada. And this work focuses on, or what I'm presenting, focuses on the contents of one chapter from this thesis, which evaluates CDOM within deposition in the oil sands region. So to provide some background, owing to an abundance of environmental sources, dissolved organic matter, or water-soluble organic matter, is ubiquitous throughout the atmospheric environment. And due to this, the deposition of organic matter is widespread throughout the planet. Now, atmospheric flux of DOM plays numerous important roles in ecosystem function. For instance, it can influence primary productivity within surface environments, and it can also impact the composition and abundance of organic matter within aquatic and terrestrial systems. Now, an important component, but lesser studied component of atmospheric organic matter is chromophoric dissolved organic matter, which is a broad category of compounds which are characterized by their ability to interact with and absorb light within the ultraviolet and visible light spectrum. Now, atmospheric CDOM presents numerous concerns For instance, it can act as cloud condensation nuclei, it can influence solar radiation budgets, and it can potentially be harmful to human and environmental health through elevated exposure. Now, the subsequent deposition of atmospheric CDOM presents another set of environmental concerns. For instance, it can alter the composition and abundance of CDOM within marine and freshwater environments, and it can also alter surface albedo, especially when deposited onto snow and ice-covered surfaces. Now, the emission sources and environmental sources of atmospheric CDOM are diverse and regionally dependent. These can be largely broken up into several categories. The first is biomass burning or combustion. A classic example is forest fires. Another is volatile and particulate emissions from biogenic emission sources, such as vegetation and forests. And another is fossil fuel combustion from anthropogenic and industrial sources. So I'm certain most of us are familiar with the Athabasca Oil Sands region, or AOSR. It's located in northern Alberta in Canada, and it's home to the world's largest known bitumen deposit. And due to this, the region has seen extensive and intensive extraction, mining, and refining processes over recent decades. Now, extensive research in the region has established that the oil sands is a substantial source of organic pollutant emissions to the surrounding airshed. These can come from a variety of emission sources, such as raw bitumen extraction, upgrading facilities, road dust fugitive emissions, vehicle exhaust, and tailings facilities, just to name a few. A variable but somewhat unconstrained fraction of these emissions will fall within the CDOM fraction. Moreover, recent work adjacent to this study established that the oil sands is a dominant source of organic aerosols rich in chromophoric organic matter. These are also referred to atmospheric brown carbon. So it seems apparent that the oil sands is a significant source of chromophoric organic matter to the airshed. However, what is largely unknown is the variability and composition of CDOM within deposition in the region. So that takes us to the objectives of this current work, which is to evaluate the spatial and temporal variation of CDOM within deposition in the oil sands region. And this will be done, or will address these objectives, through the evaluation of wet-only precipitation or wet-only deposition samples in snowpack collected in the oil sands region, and will evaluate these using optical analysis techniques, primarily absorbance and fluorescence spectroscopy. Moreover, this evaluation will characterize CDOM within deposition and identify the dominant sources contributing to deposition. So, as already established, this work involves the evaluation of wet-only deposition and snowpack samples collected throughout the region. During the meteorological year of 2021, precipitation samples collected from 3 NADP stations in the oil sands region were subsampled and sent to Trent University for optical analysis. 2 of these stations, shown above, are located in relative proximity to oil sands operations. The first of these, AB32, is located central to several major oil sands operations. This was designated as the near-field site. Next is AB36, which is located about 25 to 30 kilometers east of these major operations. This was designated as the midfield site. And the third location, located roughly 250 kilometers south of these operations, was AB34. This was designated as the background site. In addition, the samples sent to Trent University underwent optical analysis, absorbance, and fluorescence. We also analyzed them for DOC and DIC, while additional water chemistry and soluble ions were available through the NADP network and supplemented our data set. During early spring of 2023, field technicians from the Ministry of the Environment and Climate Change flew out to the oil sands region to collect snowpack samples from numerous sites throughout the broader oil sands region. Through collaboration with the ministry, 50 sites throughout this, from the sampling region, were sent to Trent University for optical analysis. In addition to this, the ministry conducted their own analysis on these snow samples and provided a range of analytes, including inorganic chemistry and organic pollutant species, including polycyclic aromatic compounds. So to briefly go over the optical analysis techniques, absorbance spectroscopy is a fundamental method which operates based on the absorbance of UV invisible light by chemical compounds within a sample medium. The absorbance measurements along a wavelength can provide information regarding the DOM composition and abundance within your sample. Additional metrics can be pulled from this absorbance spectrum to provide proxies and to provide more information regarding certain characteristics of organic matter. In this presentation, I'll be focusing on one specific metric, which is the absorbance coefficient at 254 nanometers, which is a useful proxy used in the literature to represent bulk CDOM and humic content within an aqueous sample. Absorbance or fluorescence spectroscopy is a chemically sensitive and cost-effective technique used in chemometrics, and it operates according to the principles of molecular luminescence. It's an effective method used in the evaluation of complex CDOM, and more advanced florimeters are capable of excitation and emission matrix analysis, or EEM, where your florimeter collects multiple scans of emission and excitation along a range along a wavelength range, and it produces a three-dimensional landscape of fluorescence measurements. The resulting data sets produced from EM analysis are quite information dense, and as you compile multiple EM scans in a data set, it becomes difficult to pull meaningful information from them. So that's where multiple variable analysis or modeling comes into play. One specific example is parallel factor analysis, or PARFAC, which is a trilinear approach which decomposes three-dimensional EM data arrays into individual fluorescence with significant chemical meaning. We also refer to these as fluorophores. The EM-PARFAC combination is frequently used in chemometrics to evaluate natural and anthropogenic CDOM within within complex natural systems, including atmospheric samples. Another boon to PARAFAC is that it adheres to Beer's law. So that means that we can evaluate the relative concentrations of fluorophores within our sample set. In the case of this work, we normalize our fluorescence data according to quinine sulfate units, or QSU. So at this point, we have a fairly expansive data set of numerous environmental variables for our snowpack and wet-only deposition samples. Something to note is that with the NADP data set, we convert our concentration data into volume-weighted averages. Something else to note is that the NADP stations are co-located with continuous air pollutant monitoring stations operated through a local air quality monitoring network, the Wood Buffalo Environmental Association. Several pollutants measured through these networks, including sulfur dioxide, nitrogen oxide species, total reduced sulfur, and fine particulate matter, were included in our NADP network or data set. So, looking at our wet deposition data from the NADP, when we look at our absorbance values, or absorbance coefficient at 254, we see that our values fall within the typical range of absorbance reported in the literature. However, when we look at the variation of absorbance values between stations, there's an insignificant difference between locations. When we look at the seasonal variability of absorbance measurements, we see that these values are highest during the summer months of July and August. And when we zoom in and look specific at individual weekly samples, we see absorbance peaks occur specifically during weeks that received forest fire smoke. So this may indicate that absorbance and chromophoric organic matter within the precipitation samples are influenced by wildfire smoke, particularly during the summer. So when we look at our EM and PARAFAC results, we were able to identify two significant fluorescent components. And when we compare these spectra to the literature and online databases, we were able to characterize these fluorophores. The first of them, C1, was found to be similar to highly aromatic, high molecular weight, and polymerized humic-like and fulvic-like acids. When you look into the literature, fluorophores such as this, humic fluorophores, are often identified as the prominent components of atmospheric CDOM. The second fluorescent component, C2, was similar to lower molecular weight 1 to 3 ring containing aromatic compounds. And referring to the literature again, similar fluorescence has been associated with low molecular weight amino acids such as tyrosine and tryptophan. However, there's a growing number of studies within the literature that have associated similar fluorescence with organic pollutants and PAHs. So when we look at the variability of C1 humic fluorescence in our data, we see that there's an insignificant difference in fluorescence between our locations. This is similar to what we see with the absorbance. Moreover, when we look at the seasonal variability of C1 fluorescence, we see that it's elevated during the summer months of July and August. And then again, when we look at our individual weekly samples, we see peak fluorescence during those wildfire smoke weeks. So this may also indicate that humic fluorescence is influenced by biomass combustion. Moving on to that lower molecular weight fluorescent component 2, we see that there is a significant difference between sites, where fluorescence is elevated at the near to midfield locations, so AB32 and AB36, and fluorescence is often below detection at our far field site at AB34. This may be an indication that C2 fluorescence is influenced partially by oil sands emissions or oil sands processes. And when we look at the seasonal variability of C2, we see that it's elevated during the colder winter months. So when we include the additional environmental variables with the NADP network or NADP data set, we can further characterize these fluorophores. And when we look at C1 humic acid fluorescence, we see that it's positively correlated with ammonium, dissolved organic carbon, and fine particulate matter. And when we conduct principal component analysis on this data set, we see that C1 humics are positively correlated with principal component 2, which has high variable loadings of fine particulate matter and ammonium. Considering that these variables can be influenced and are associated with forest fire emissions and biomass combustion, this may be further evidence indicating that C1 humics are influenced by forest fire emissions. Moving on to C2, lower molecular weight fluorescence, we see that it has a positive correlation with calcium, magnesium, sodium, chloride, NOx species, and total reduced sulfur. When we look at our principal component analysis again, we see that it's positively associated with principal component 1, which has high loadings of calcium, magnesium, and sodium. And we know that the oil sands is a substantial source of base cations, emissions, and deposition to the region. So this may be an indication that C2 fluorescence is influenced by industrial sources. Moreover, the strong agreement of C2 fluorescence with NOx species could further suggest that this fluorescence is associated with diesel mining emissions from the oil sands fleet, mining fleet. Moving on to our snowpack data, when we look at our absorbance measurements, the absorbance at 254, it is elevated at sites near to oil sands operations and generally decreases or is lower at further field locations. This is contrary to what we see in the NADP wet-only deposition data, which presented limited difference between our locations. Moreover, when we conduct EM and parafac analysis on the snowpack data, we identified two distinct fluorescent components. And interestingly, the spectra from these fluorescent components are similar to what we see in the NADP data, or in the NADP precipitation samples, where the first fluorescent component is similar to humic acids, and the second fluorescent component is representative of lower molecular weight compounds. And when we look at the variability of the humic acid fluorescent component, in this case, we see that it's elevated at the near field sites close to oil sands operations, and it's generally lower at further field locations. Once again, like our absorbance, this is contrary to what we see in the NADP precipitation samples. And then finally, looking at C2 lower molecular weight fluorescence, we see another spatial gradient, or similar spatial gradient of elevated fluorescence at near field industrial sites with rapid decline with distance from oil sands operations. So what this seems to be an indication of, or maybe indicative of, is that the oil sands is a dominant source or key source of bulk CDOM deposition, at least during the winter. And unfortunately, at this time, we don't have our hands on the additional chemistry data through the Ministry of the Environment and Climate Change, but we eagerly await that so we can further characterize these fluorophores within the snowpack. So what are the major takeaways so far? It seems that the oil sands contributes to the deposition of lower molecular weight 1 to 3 ring containing fluorescent compounds, so C2 fluorescence throughout the oil sands region. However, the sources of humic CDOM, as indicated by absorbance, and C1 fluorescence is mixed. And this is seen through the high fire smoke dominance or influence in the wet-only NADP samples during the summer months, but the clear influence or the likely influence of oil sands emissions on bulk snowpack deposition during the winter. So further analysis and further validation is required. However, considering the cost-effective nature of absorbance in fluorescence spectroscopy, these techniques may be viable alternative methods for evaluating organic matter deposition in the oil sands region, or maybe a viable supplementary method. Yeah, so that's what I have so far. Any questions?
Any questions for Dane? I think we have time for one.
What an amazing data set. Do you have any insight onto how much of the dissolved organic carbon that you're measured in your deposition samples and what fraction of that is chromophoric, as you were talking about? It might not be easy to determine that.
Yeah, with the current state of the data that I have right now, it's difficult to say what the actual fraction is. But chromophoric organic matter can represent a fairly substantial portion of organic aerosols within the atmosphere. Some papers have reported it up to 70% of the water-soluble organic aerosol fraction. But in the case of the oil sands, it's pretty hard for me to say. I do have I have some measurements of dissolved organic carbon within the sample set, so I can compare them, but once again, it's difficult to say how large that fraction is.
And if I got time real quick, also, do you see any changes in your absorbances versus precipitation pH that can sometimes change how things absorb?
Yeah, there's the question of the influence of pH conditions on absorbance and fluorescence. It's an ongoing issue in chemometrics and optical analysis. I haven't seen that strong of a relationship in the data that I have, but it's something that you have to be cognizant of that quenching effect, right? That's the best answer I can give, yeah.
Thank you.
Yeah.
Thank you very much, Dane.
Yeah.
Appreciate that. Let's talk. Next up. University of Mexico.
Thanks, Winston. Good afternoon, everyone. I am going to present a study about wet atmospheric deposition. We are working for more than 20 years in Mexico as a country, basically in two regions, in the Mexico City metropolitan area and the region of the Gulf of Mexico. I would like to acknowledge all my staff that is working at this moment at the laboratory and some people that is working surrounding areas of Mexico City and in other regions like the Gulf of Mexico. We understand that we only can to do this kind of research with the collaboration of different institutions. I appreciate all the help from institutions here in the United States, EPA, NADP, and USGS regarding the comparison laboratory programs, Evan Reed for the World Meteorological Organization, and several years with Van Bowerson. And thanks to Gregory and Noel that they are collaborating with us. We are very happy to participate in this program about intercomparison laboratory. What happened with the Mexico City and the different studies that have been developed there? Mexico City is a very nice laboratory in order to do the research regarding atmospheric pollution. We are more than 20 million inhabitants. And air quality in this area began the study during the 70s. The first studies about air quality. And the first efforts began in the 80s. And using protocols for the World Meteorological Organization and the National Atmospheric Deposition Program was in the date of 2008 when we began to use this kind of protocols. This is the reason why I want to present some information about it. Here is the Mexico, the country. Here appears the Mexico City metropolitan area in the center of our country. And another very interesting region, because it's an international importance, is the region of the Gulf of Mexico. Regarding Mexico City metropolitan area, we need to understand what happened throughout the years. During the 80s, the most important pollutants were SO2, total suspended particles, and lead. And this bad air quality paid attention in the people and in the people that take the decision and change the lead in gasoline, reduce the use of lead, and the fuels that were consumed at the Mexico City metropolitan area changed from fuel oil to natural gas. Good decision regarding these pollutants, SO2, TSP, and lead. Both increase the ozone precursors, nitrogen oxide, nitrogen oxides and volatile organic compounds. And then at this moment we have the problem of ozone, our quality, about our quality regarding ozone, PM10 and PM2.5, and acid rain problem continues in Mexico City. All the results that have been obtained for several decades has been resumed in this publication. It's nitrogen and sulfur compounds in ambient air and in wet atmospheric deposition at Mexico City metropolitan area, with the collaboration of the NADP, the big guys, one of the co-authors. And the most important findings were the control strategies applied at Mexico City have achieved an important improvement in SO2 air quality. Good news. However, sulfate continues being the more important ions related with the acid rain precursors. And if we observe the air quality during the 80s until now, there is a great reduction of emissions, and there is a result in the air quality. And the air quality standard in Mexico City is in attainment. At this moment, it's in attainment area regarding SO2. However, as you can see, the sulfate is the most important ion and is over the nitrate values. What happened regarding the pH? We have been developed this kind of maps and box plot for all the ions. However, for this presentation, I am presenting here the pH values. The more acidic values are in the south of the Mexico City metropolitan area. The dominant winds come from the north to the south. These are the mountains. And the sampling size are ordered regarding the altitude. And the more acidic values are in the south, in the area with mountains and ice places. And the urban area is here, and the industrial area is located at the north. Regarding with the influence of the winds, the industrial has a very important impact in the city. The sampling size that I already showed, there are the sampling sites for the Mexican government, 16 sampling sites that collect the samples weekly. At the University of Mexico campus, we collect the samples daily and weekly. If we need to correlate the wind direction with the result of the chemical of the wet atmospheric deposition, it's not easy to collect weekly because you receive a lot of events. In the case of the Mexico City, when we are collecting weekly and daily, we analyze the daily events and relate it with the wind direction. All the events that present a pH below 5.6 come from the north, and the events that present the pH above 5.6 comes from the south. This is then the same line that we are observing here, that the pH, more acidic values are located here at the south. What happened with reactive nitrogen? This is a very nice issue in this moment. And we can relate and use the ratio, the ammonia to nitrate in the same sampling sites. And you can observe that the median is above 2. In this case, the reduced form of nitrogen in ions, ammonia, is above the nitrate. The results are similar to some cities in the United States. Several years ago, I remember we made a comparison with the area of Boulder, Denver, are in the same level. This is why it's necessary to study the reduced form of nitrogen. Like in another sites, in ambient air is evaluated regularly in nitrogen oxides. This is the oxidized form of nitrogen. with what happened with ammonia. Ammonia is not measured in a regular way. But if we analyze the radio, we need to put attention in this evaluation in ambient air. This is a recommendation for the future. Inside Mexico City, there is the campus of the university here. And inside the campus is a small urban area, urban reserve, natural urban reserve. And in this area, we made an experiment to evaluate the chemical composition of the rain. We are in the same line that the other sampling site in Mexico City. Ammonium present the high levels, then sulfate, calcium, and you can observe that sulfate is higher than nitrate, and in this case, ammonia is higher than sulfate. Reactive nitrogen is very important to study, and a great difference comparing with some places in the United States. So in the United States, most of the places of sampling sites present sulfate very, very, very low. The problem is that in Mexico, we continue using fuel oil, not in Mexico City, but outside Mexico City. There is an influence from the emission sources that are located upwind Mexico City metropolitan area. Another very interesting region to study, I always talk about the international region because United States, Cuba, and Mexico, we are part of this. And if we analyze the transport from the Caribbean area to the Gulf of Mexico, Belize is another important country that we need to take in consideration. And the most important findings in the research was about the Gulf of Mexico region has important sources of acid rain precursors. We know the pH values at Mexican sampling sites are very similar to the NADP sites. At the Mexican sites, the radio SO2 and NO3 it was higher than in the United States. Acid deposition has a major contribution for the SO2 emission, comparing with nitrogen oxides. This is the only site that has been working in Mexico for more than 20 years. In Spanish, it's La Mancha. La Mancha is the name. It's located in the port, closer to the port of Veracruz in Mexico. In order to have an impact, an international impact, presenting an article in the area of the Gulf of Mexico, we need to collaborate with the NADP and uses the NADP sites from Texas to Florida, Louisiana, and etc. And the results are very, very important because the influence of the SO2 and nitrate radio in the area of La Mancha will represent a very nice result, and we need to give this information to the people that take the decision. We need to change the fuel, or maybe to apply engineering strategies to reduce the sulfur content in this fuel. Part of the group, when we are collaborating, more of the people are chemical engineering, biologists, and it's very important the people that work in meteorology. Analyzing using the high split, this is the transport behavior during the rainy seasons. This is the site that we have in Veracruz. This is using the back trajectories of the high split during the rainy seasons. Comes from the Caribbean area, cruise the Yucatan Peninsula. Yucatan Peninsula is plain, no mountains. Then cruise again the Gulf of Mexico and go to the Veracruz, the coast. All this area, United States and Mexico, have a lot of emission sources, offshore oil plants are here. And what happened during the dry season, the transport comes from the northeast, And if we analyze that in the United States there are a lot of sampling sites, and in Mexico we only had one, we need to increase the number of sampling sites. We are collaborating with the people of Campeche. We have information from 3 years ago, but we need to increase the sampling site in the Caribbean area, and why not in another sampling site on the coast of Mexico. This is a research that is in review. It's for one of my PhD students. This is about the reactive nitrogen in the area of the Gulf of Mexico. This is all the information that you have of NADP for wet atmospheric deposition, GasNet for nitrogen dioxide. And in the area of Mexico, we have 2 sampling sites in these years from 2017 to 2020. 2 sampling sites, one at La Mancha, and the other in the Port of Veracruz. Both the Port of Veracruz only works our sampling site for 4 years. And these another 3 air quality monitoring stations are located in the near Poza Rica, La Mancha, Minatitlan. And this air quality information is very important for us because we are working with the government of this state, and they are increasing the number of air quality monitoring stations. This is the Veracruz state, and these are the new air quality monitoring stations. There are 10 sampling sites, and the idea is that these air quality monitoring stations collect at the same time wet atmospheric deposition, and why not use passive sampler to evaluate nitrogen oxides, ammonia, and mercury. And what's next? You know, I have already talking about, it's very important international collaboration. For several years ago, I have been here in NADP meetings. I have been with you working, collaborating, and thinking what we can do at this moment. I know that all of you, and of course, people like in Mexico, we have different problems, economical problems, and we need to think of how to save the money. Both the collaboration is a very great opportunity to maintain our research in different areas that are very important. This is important for us. Atmospheric deposition is very important to evaluate atmospheric deposition in urban areas. For several years, I have been in touch with all of you, with Greg Viterbe, talking about city depth. Why not to increase the study in United States, Canada, and Mexico, and etc. Mexico metropolitan areas and another urbans in North America. One of our students developed a thesis comparing wet atmospheric deposition of urban area of North America. We in Mexico, we need to learn about the good experience and the bad experience that you have already developed here in the United States. Reactive nitrogen evaluation. Unfortunately, most of the pollutants that the people in Mexico and the government are interested to evaluate are criteria pollutants, because the criteria pollutants are regulated. Part of the reactive nitrogen evaluation corresponds at that NO2 is a criteria pollutant, so the analyzer can evaluate NO2 and NOx. But the importance of reactive nitrogen, we need to put the idea in the mind of the people that is taking the decision. Now, include ammonia measurements in ambient air to close the circle, and evaluation of sulfur, nitrogen, and also critical loads. Mexico City, so you can see my first slides, we continue with the problem of ozone. regarding to the Mexican ozone air quality standard. We are non-attainment area. But we need to pay attention in the future to work in critical loads, and of course, sulfur and nitrogen. Then a new project is evaluation of atmospheric mercury. This is part of our good result of the collaboration with NADP. I appreciate the help of David Guy, of Bit Grande, Waya Sherlock, in one moment, Mark Olson. And this is the equipment. This is a reality that is operating now at the university campus in Mexico City. But about the whole of Mexico region, here appears the air quality monitoring station for the government. We need to improve in this area emission inventories, including all the industry. You know all the industry, movement of ships, coastal cities, all industry. Why not implement passive sampler for ammonia, for mercury, in a specific air quality and atmospheric deposition sampling site that at the moment already exists. As you know, we have a lot of work to do. I am here to receive some kind of good advices and continue working in this field. But I will be happy if a lot of people from Mexico would like to come here at the NADP. The people that is working at the government, the people that's taking the decision, they need to be here, where the people knows what happened in the atmosphere and with the wet atmospheric deposition. I appreciate all the suggestions and comments of the National Atmospheric Deposition Program. And I am presenting here the rope of the University of Mexico, where is located the mercury analyzer. Here is the other side that is part of the government facilities that is located at the north of the city, where the atmospheric deposition for mercury would be developed. And NADP, UCPA, and the University of Mexico, we are collaborating in a project to develop a campaign for the next year. Dr. John Walker has been in Mexico with a Picarro instrument and comparing with some passive sampler that we are preparing at Mexico. And thank you very much for your attention. Are there any questions? Okay.
I think we have time for one quick question.
All was clear?
Rodolfo, well, I have a question for you. I was really struck by the time series plot that you had of sulfur dioxide concentrations in the Mexico City region and that tremendous drop in the early 1990s. Can you comment on a similar time series or concentration trends for NOx along that same time series?
Yes. During the 80s, we had a problem of sulfur dioxide.
Right.
We are non-attainment area regarding sulfur dioxide. Total suspended particles, no PM10, no PM2.5 in this moment, and lead, because use lead tetraethyl in gasoline, has anti-knocking. And in this moment, Mexican government apply strategies. Reduce the lead in gasoline would increase the reactive hydrocarbons. Was one. But what happened regarding CO2 and particles? The fuel that was used at the power plants inside Mexico City was bunker oil, fuel oil, and then was switching to natural gas. Good regarding CO2, good regarding particles, but increased nitrogen oxides. And remember, if you use natural gas, now the problem is not only criteria pollutants. You maintain the same levels of carbon dioxide. The natural gas continue being a problem. Okay. But this is here the you need to measure in order to evaluate what happened in the atmosphere, not only imagination. Okay? Okay? Thank you very much.
Okay, next up, we have Anam Khan from right here in Madison.
Hey, hi everyone. I'm Anam, and I will be presenting some initial data that I've been collecting as a part of my PhD over this past summer in Central Illinois, monitoring ozone fluxes at an eddy covariance tower at the agricultural field. So I want to start with an overview of the dry deposition of ozone onto the land surface. And the big thing to focus on here is this big green box, which shows all, oh, okay, you probably can't see my mouse. Well, the big green box in the middle, which shows the different components of dry deposition of ozone onto the land. There has been a lot of literature, I've only listed some here, that has really demonstrated that we can measure this bulk flux of ozone to the land surface at eddy covariance towers. And a good chunk of the literature has argued that we should be doing this. To parse well, it's demonstrated that these flux measurements have a great utility in evaluating the dry deposition of ozone in chemical transport and regional air quality models. And then from an ecosystem perspective, many have also argued that it's this red box right here that shows ozone uptake by stomata that is the critical flux to start monitoring or estimating, because that's what really determines the phytotoxic dose that's delivered to the plant, as opposed to ambient concentrations. So motivated by much of the literature, the fact that it is demonstrated that it can be done and that there's great utility in these flux measurements, we did just that using UV absorption-based instruments at an eddy covariance tower that measures high-frequency wind speeds and a lot of other micromet variables. And we were comparing high-frequency UV absorption-based instruments, which have not been widely available or used in ozone flux measurements. And so we compared two instruments. The first one is the NASA Rapid Ozone Experiment developed by Hanoun and colleagues. And we compared ROSE with a widely available commercial instrument from 2B Technologies that had an experimental upgrade to collect data at a faster frequency. So the question with the 2B Technologies was that, is the higher frequency that it's collecting data at really capturing the turbulent fluxes of ozone? And since ROSE has been validated with a chemiluminescence instrument before, we're kind of using ROSE as a standard, but also going back and forth to see what we can get from each of these systems. In the second part of my talk, I'll present how I partitioned. I'm using two methods, different methods to partition this bulk flux that this entire green box contains into this component that I'm very interested in, which is ozone uptake by stomata. And so utilizing all of the other flux measurements at the eddy covariance towers, like the exchange of CO2 and water vapor, we can use some established methods to try to parse out that stomatal component and estimate that fraction. And then I'm comparing that with another method that was developed to parse out the transpiration flux from the combined ET flux, and using that to also get the ozone stomatal flux using the 10 hertz raw data. So I'll go over that as a second part. So we did this at a site near Bonneville, Illinois. It's a cornfield site. Just a quick summary of this site. We had the instruments up from early June to about late September, I'll be presenting data from July, initial data and in very initial estimates from July. During this time, it's a very sunny site. It saw it's rain-fed agriculture, so there was sporadic rainfall during this entire season, and fluctuations in soil moisture. And then during the data that I'm presenting saw a little bit of a decline in atmospheric dryness and vapor pressure deficit. The local EPA monitor near the site, these are the long-term trends on the left here from 2011 to 2023, and they just to show that during our season, the site can reach above 40 parts per billion for the 8-hour maximum. So the two instruments that we compared, the NASA ROSE has shown good agreement with the chemiluminescence system. Previously, when it was flying on a NASA airplane, it is built for higher sensitivity of ozone, also has a very high flow rate to be able to flush this cell that you're seeing where the light is attenuated due to the ozone sampling. So that cell gets flushed out rapidly due to the higher flow rate, and so it's able to capture a 10 hertz signal. And the 2B technology's dual beam ozone monitor is typically used as a slow response monitor. The company suggested that there is an eddy covariance upgrade that they can make, but it needs to be tested. So that is exactly what we did, because that was highly, highly uncertain whether the 2B is actually able to capture the turbulent fluxes, even if it's logging data at 10 hertz. Okay, so for the very initial comparison of the fluxes between the 2B and the ROSE, I'm going to focus on the figure on the right first, which is the diurnal mean cycle for July. And while the 2B and the ROSE produce similar diurnal cycles, so 2B can see the net flux towards the land, which is in negatives here. There's a lot of disagreement in the magnitude. So the green line is just 2B that didn't see any more processing after collecting the data. And then the blue and the yellow lines are the 2B data that I put through some despiking to remove some noise initially, and then a little bit of a smoothing with filling in those noisy spaces with a 6-second rolling mean or a diurnal mean, so the mean at that exact point during July. And so the bottom plot here on the left kind of shows a disagreement between them, just the differences. And there are large differences between 2B and ROSE, even though the patterns seem similar. But processing the 2B, a little bit of the noise removal decreases the disagreements during certain times, like in late August, but not too much. And so that's like a very initial first comparison of the data from this past summer. So because the diurnal cycle seemed reasonable from 2B, the real question really became about whether these fluxes whether these patterns are reasonable. Like, what is a 10 hertz data saying? So I'm going to present two figures, the first one being the lag covariance between vertical wind and ozone concentrations for half hours worth of data. So the multiple lines here show different half hours within the July data. And these are some of the half hours that showed some of the highest fluxes. So they are probably some of the most highest quality half hours. And so where you see that sharp peak in the lag covariance, that's from the NASA ROSE instrument. And the 2B model is the one that has a lot of noise in its lag covariance. And so this plot first suggests that the 2B is largely, it's really, really hard to tell whether the covariances it's detecting at any half hour are meaningful, or if it's a lot of noise, because the rose has a very clean lag at a very clean peak in covariance at a defined lag, which is what we should be seeing. So that means that as we shift the ozone time series around, we don't see covariance with vertical wind except at this very reliable shift. And so that's a very clear and clean signal of a half-hour covariance that is related to turbulent transport rather than noise. So moving a little bit deeper into analyzing the 10 Hertz data, this is also, again, for multiple half hours in our time series, the co-spectrum between vertical wind and ozone. And from NASA ROSE on my left, and then the noisy one on the right with the 2B. And so with NASA, the ROSE instrument, again, I saw a much more cleaner signal in terms of translating the time series into the frequency space, and then seeing that at the lower frequencies, there's a very sharp peak in, there's a very clear frequency that's contributing to most of the covariance that we're seeing at the half hour. And then we see a decay at the higher frequencies, which is something that we often see with H2O, like water vapor or CO2 as well, and is really telling of being able to capture the eddies that bring most of the flux. Whereas the 2B, there's a very, very faint peak at those really important flux-carrying eddies. But again, it is very noisy. And after despiking, I mean, maybe it's just my eyes. It got a little tiny bit clearer, but still a lot of noise. And so I'm going to present the stomatal component now from the NASA ROSE data, just based on these signals. I'm still working with the 2B data a lot more. I know there's a lot of literature on these frequency loss corrections, and so I'll look into that. But for the stomatal component for today, we're going to move on with just the NASA ROSE data. And so I'm going to explain the first method that I tried for isolating the stomatal component, and then a second one. So first, I just want to start by saying that the deposition velocity of ozone we measure because we measure the flux and the concentration, and the deposition velocity is just flux over concentration. And that deposition velocity that then can be broken down into the aerodynamic resistance, the boundary layer quasi-laminar resistance, and a bulk surface resistance called the canopy resistance to ozone. So chemical transport models break their canopy component, the RC component, into many different component resistances that operate in parallel or series, but we can't do all of that yet, or without introducing a lot of heavy-duty modeling into eddy covariance data. So we can calculate RC as the leftover in the deposition velocity after we've calculated aerodynamic resistance and boundary layer resistance. And then from the canopy resistance, we could use some established methods that have estimated stomatal conductance or canopy level stomatal conductance to kind of also estimate a stomatal component out of that canopy component. But here, all I'm showing is that the deposition velocity at this site is mostly dominated by the land component, the RC component, throughout most of July. And so honing in on that stomatal component, and GS is just the inverse of RST. So if you ever read papers about estimating the stomatal component from eddy covariance data of ozone dry deposition, you'll probably see Penman-Monteith inversion on almost all the papers. So it's a very widely used method to use the ET flux from the tower and invert the Penman-Monteith equation to get a canopy conductance to water vapor. The tower water fluxes are the full ET flux, so it requires an assumption and narrowing down to conditions when transpiration dominates, and so the Penman-Monteith inversion becomes a little more applicable. And that's how it's typically done. Recently, there have been a few papers that will say that, okay, well, we're also collecting carbon exchange, which we can get gross primary productivity from, and we can start using these along with the water vapor flux to fit a GPP-based stomatal conductance model to get its parameters, fit it to the Penman-Monteith inversion of H2O flux-derived stomatal conductance. And so that's the first method, is what I've done with our data, is just fit a leaf-level stomatal conductance model from Medlin et al. To the GS estimates from the PM inversion from H2O flux, kind of coupling both the CO2 and H2O flux at the tower to derive the ozone stomatal component. The second method, which the results are incredibly initial, but I'm trying to extend conditional eddy covariance to isolate the stomatal fluxes in ozone dry deposition. And conditional eddy covariance just takes traditional eddy covariance and says, we can parse out the transpiration component of ET by looking for eddies in the 10 Hertz data that are characteristic of stomatal sources versus non-stomatal sources. So it puts a lot of conditions to kind of come up with an eddy covariance that is just based on those eddies to try to get the transpiration separate from evaporation, soil evaporation. And so this is just a comparison of the stomatal component of ozone deposition velocity using the Penman-Monteith and Medlin approach, which is on my right. It's the figure with the lower green flux. The green flux is the stomatal flux. And so Penman-Monteith and Medlin combination kind of suggests that there is a non-negligible, non-stomatal component at this field, and so does the conditional eddy covariance one. And so that means that the ozone flux that's coming down isn't all taken up by stomata or isn't all taken up by the corn, that there is like a non-stomatal destruction component here. And it's just the two methods differ greatly right now about, one is really saying that the stomatal component is pretty dominant, and then the other is saying that the Medlin et al., where the green is lower than the blue in the mornings, in the afternoons, is saying that, for me at least right now, is that the morning flux is dominated by the stomatal flux, but the afternoon, the non-stomatal component becomes more important in ozone dry deposition. And so just some very initial conclusions, and these are a lot like, I saw this, but this. And so for the instrument comparison, the 2B showed similar diurnal patterns, but looking more into the 10 Hertz data, the expected signals that you expect from good flux is faint, if even present, and despiking doesn't really change that. And the ozone partitioning, something that I could definitively say as of right now is that there is a non-stomatal component, so the plants aren't just taking up all the ozone that is dry deposited onto the land. But there is disagreement between the partitioning methods I'm using about the magnitude and the fraction of the non-stomatal component. These are my references. Thank you.
Thank you. We have time, I think, for one very quick question.
Great talk. You're definitely trying to work out some stuff that's never been worked out before, so that's great. Is there anybody making any kind of actual measurements on the plants that you can check against to see what the actual stomatal conductance might do over the course of a day, and then compare that with what you're seeing in the eddy covariance?
Yeah, thank you. I'm not aware of leaf level stomatal conductance measurements, although the site is a huge operation with Lisa Ainsworth and Carl Bernacchi, and I know Lisa does a lot of leaf level work, so I would not be surprised if there are leaf level diurnal measurements, which would be amazing to check against, but I am not aware, or they're not available to me as of yet. Yeah, thank you.
Okay. Thank you very much. Thank you. Okay. Next up, we have Jian Feng from Environment and Climate Change Canada.
Good afternoon, everyone. My name is Jian Feng from Environment and Climate Change Canada. I'm a physical scientist. Today I'm presenting long-term trends and causes of springtime maximum of ground-level ozone in the eastern US and Canada during 1990 to 2016. I would like to acknowledge the contribution to this study by my colleagues, Maria McDonald, Li Ming Zhang, Jason O'Brien, and Irene Chen. There are two objectives for this study. First, we would like to derive the long-term trend of ground-level ozone in the eastern US and Canada during 1990 to 2016. Specifically, we would like to know how diurnal cycle of ground-level ozone changed for different seasons in response to emission reduction of NOx and VOCs. Second, we would like to investigate possible causes for the springtime maximum of ground-level ozone in the eastern US and Canada. Due to time limitation for this talk, I will mainly focus on the second one. So how ground-level ozone is produced? Basically, it's formed by photochemical reactions involving nitrogen oxides, VOCs, and carbon dioxide. Stratospheric ozone can also be brought to the surface. This is commonly known as stratospheric ozone intrusion. There are three major components for ground-level ozone production, namely VOC, NOx, and solar radiation with wavelengths less than 400 nanometers. If any component is missing, production of ozone will stop. The data used in this study for ground-level ozone, we used measurement at rural sites by the CASNET for the US and from Capmont for Canada. We also use leaf area index derived from all this satellite data. For this study, we grouped the measurement site into 4 subregions, mainly following our previous 2 studies. These 4 subregions, Region 1, Eastern Canada and Northeast of US, Region 2, Middle-West, Region 3, Mid-Atlantic, and Region 4 is the Southeast of the US. Because emission reduction of NOx can cause ground-level ozone either increase or decrease depending on the ratio of VOC to NOx and availability of solar radiation. So we would like to see how Dyno cycle of ground level ozone changed in response to emission reduction of NOx and VOC. For this purpose, we compared the Dyno cycle of 2 periods, the first 10 years versus the last 7 years. Because when this analysis was carried out, we only have data up to 2016. In future, we will update this analysis up to 2019. We will do this comparison for different months and for different percentile from 10 tenths to P95. Because the change in Dyno cycle from May to September and November to March was similar, so we group these 2 5 months. The first one is a warm season, and the second one is a cold season. Now we take a look at changes in Dyno cycle of ground level ozone. The last 7 years versus the first 10 years. As you can see, for May to September, the ozone concentration reduced consistently for all hours. Of course, it's more pronounced for P95 and P75. For cold season, November to March, except for P95, ozone concentration increased for all hours, both day and nighttime. For April and October, except for P95, we see that there's a little change in Dyno cycle. Now I move to another topic, springtime maximum of ground level ozone. So what is springtime maximum of ground level ozone? You may ask this question. So although May to September is considered as ozone season, at many middle latitude sites, ozone concentration peaks in spring instead of summer months. This is commonly known as springtime maximum of ground level ozone. In this two graph, shows the Dyno variation for different months in 1990. For the site in Northeast, we can see ozone concentration peaks in April, while for the site in Middle Atlantic, ozone concentration peaks in July. The reason for springtime maximum 20 years ago, basically stratospheric ozone intrusion was once proposed as a mechanism for springtime maximum. But a study later on generally excluded this possibility. Recent assessment of this issue indicate that this is still an open question. There are no definite and conclusive explanation for springtime maximum. So we are trying to, for this study, we are trying to shed some light to this long-term open question. So we took a relatively different approach to this question. We look at the time series of 10-year composite. In this two graph, it shows the time series of Dyno maximum and Dyno minimum for the first 10 years and the last 7 years. for the time series, we applied a digital filter, KZA filter, to smooth out high-frequency variation. From these two graphs, I would like to mention, despite the dino maximum decreased significantly in 27 years, dino minimum actually changed very little in over 20 years. Now we focus on 1990. I forgot to mention, this graph shows that time series for one specific site, ARE 128. It's a site, Castanet site in Middle Atlantic region. From this, for this graph, if we focus on blue line, the solid blue line is the time series of daily mean. The dashed blue line is the average of dino maximum and dino minimum. So we can see on average in terms 10-year composite, daily mean can be approximated by the average of dino maximum and dino minimum. We define dino ozone protection as the difference between dino maximum and dino minimum. Then we can approximate the dino mean as the sum of dino minimum and half of dino ozone protection. Because dino ozone protection has a relatively small impact on ozone dino minimum, as we saw from previous slide. So these two variables basically can be treated as independent variable. So now we take a look at the time series of smooth 10-year composite of dino maximum and minimum of ground level ozone in two regions, Middle Atlantic of the US and Canada. First, I would like to mention for both period, the first 10 years and the last 7 years, and for both regions, there's a common feature, that is dino minimum increase from January and decrease from May to September. This is a very important feature in our explanation. If we focus on Middle Atlantic and we compare the last 7 years to the first 10 years, we can see that dino minimum increased during January to April and decreased during May to September, but the magnitude was relatively small. However, if we look at dino ozone production during May to September, it decreased by a much larger magnitude. So the net effect of these two trends is dino mean ozone concentration peaked in April during the last 7 years, while during the first 10 years, it peaked in July. So here's the question: what causes the dino minimum to increase from January to April and a decrease from May to September? To answer this question, we look at the monthly dino variation of ground level ozone normalized by dino value at 18 local time. The reason we normalize the dino variation by dino value at 18 local time is because after 18 local time, UV radiation is limited, and the production of ozone is limited, and it stops during nighttime. So basically, after 18 local time, the ozone dino cycle entered a decay process. From this graph, here the black line is the variation for April, and the blue and red line are for May August. So we can see normalized ozone mix ratio decreased much faster in May to September than in April. So a decay process can be described by a differential equation. Analyzing this differential equation, we proposed a new variable, DP. DP is ozone diurnal decrease coefficient, and it's defined as ozone decrease rate between 18 and 6 local time, normalized by the diurnal value at 18. Now we provide a conceptual explanation why ground level ozone diurnal minimum increased from January to April and a decrease from May to September. We look at DP, our two value. One is our low value and another is our high value. And we look at the ozone diurnal cycle. Starting from 6 p.m., ozone concentration increase and peaks at 3 p.m. From 6 a.m., ozone entered a decay process. If we have our DP, our low value, ozone concentration at 6 p.m. the next day will be higher than ozone concentration at 6 p.m. at the previous day. If otherwise, if DP is our high value, ozone concentration at 6 p.m. the next day will be lower than previous day. So as we see, from January to April, DP is our low value, and from May to September, DP is our high value. Therefore, from January to April, we saw the diurnal minimum increase, and from May to September, ozone diurnal minimum decrease. So now comes to our explanation. There are 4 points, one step by step. I use April and June to represent the spring and the summer months. First, we saw that ozone diurnal decrease coefficient in April is much lower than June. Second, due to higher ozone decrease diurnal decrease coefficient, ozone diurnal minimum decreased from May to September. In terms of ozone production, generally it's higher in June than in April. Then we consider the combination of these two effects. When diurnal ozone production is significantly higher in June than April, it compensates for higher decrease coefficient in June, so monthly concentration peaks in June. And this is the case for 1990 in Midwest and Mid-Atlantic. With the reduced emission of VOC and NOx, the ozone diurnal production is much reduced, and it cannot continue to compensate the higher ozone diurnal decrease coefficient. So monthly concentration of ozone peaked in April in Mid-Atlantic and became comparable in May and June in Midwest nowadays. So now comes to the last question: why diurnal ozone decrease coefficient much higher during May to September than April? To answer this question, we look at the seasonal variation of DP. We found that seasonal variation of DP is very highly correlated with leaf area index. The mean value of correlation coefficient for all sites in the East, US, and Canada is 0.93. So this is the last slide. Now comes to our hub size. we propose that high leaf area index during summer months increase ozone dry deposition and therefore increase cause DP much higher values in summertime. There may be some other factors and play a role in high value of DP, such as seasonal variation of nighttime, NOx titration, temperature, or other chemical reactions. But at this time, we cannot either confirm or exclude. For future work, we would like to perform some sensitivity test study using GMAC to see how seasonal variation or leaf area index impact the time series or ozone diurnal minimum. We also would like to do some evaluation of model output with derived correlation between DP and ALI. So I stop here. Thank you for your attention.
Thank you. In the interest of time, one short question. I think we're running a little over.
Thank you. Thanks. This is really interesting and a great talk. I've seen, modeling studies that suggested, in the future there may be a reversal of the ozone seasonal cycle, and so it's really cool to see this presented in the measurements. And so I was wondering, if you've, that paper had sort of suggested there could also be a role for rising methane as well as climate, and I'm wondering if you have considered or would be interested to consider possibly those variables as well.
Sorry, and could you repeat the last question?
Sorry, it's supposed to be a quick question and it took a really long time. Have you considered a possible role for rising methane in terms of just having background precursors, or also possibly climate in terms of the seasonal cycle that you're seeing? Thanks.
In terms of seasonal variation?
In terms of, so you're showing essentially higher ozone in towards the winter, right? Which is different than what it has been historically, or towards like the ends of the year as opposed to in the summer. No?
Okay. I'm sorry.
We might want to do this
that.
offline.
I'm sorry. Maybe I didn't catch your question, and maybe we can talk later on. Okay, no problem. Thank you.
Thank you. Finally, our last presentation will be done remotely, given by Meg Gustin of University of Nevada at Reno.
Perfect. Thank you very much. So I know you folks are going over, so I'll try to make this quick. What I want to do is talk about dry deposition, measuring dry deposition of mercury-2 using arrowhead samplers. And this work has just been recently published in a paper in Science of the Total Environment. And here are the co-authors on the paper. Can you go to the next slide, please? So in order to really understand mercury deposition, we need to measure wet deposition and dry deposition. And to measure wet deposition, they measure concentrations of mercury in precipitation and the volume of precipitation. But dry deposition is a little bit harder to get at. So you can measure dry deposition in liter fall, and that will get at GEM, gaseous elemental mercury. But then surrogate surfaces are something that can be used to directly measure reactive mercury, which would be gaseous oxidized mercury and particulate bound mercury, or just gaseous oxidized mercury. And there's been multiple methods that have been developed to do this. And I've just listed ones. Some measure reactive mercury, some measure gaseous oxidized mercury. But what I want to focus on is a method that we developed and first published a paper on in 2009. Next slide. Oh yeah, oops, go back up there. Sorry. I just wanted to point out on this slide that a good dry deposition sampler should reflect the turbulence and the deposition velocity of the compounds that are being deposited. Next slide. So this is what the arrowhead sampler looks like. It's an aerodynamic polyoxymethylene disc. And if you look at it, I can't point, but there you'll see in the middle, there are two circles. The outer circle is a drip shield to prevent like condensation going on the membrane or light rain events affecting the membrane. And then the membrane is the lighter circle in the middle. And it's a cation exchange membrane, and placed upside down, it's used to capture gaseous oxidized mercury. So, and then the deposition is calculated based on the mass of the mercury on the membrane, subtracting the blank membrane, the area of the membrane, which isn't on there, and then time. Next slide, please. So I just wanted to point out, what the sampler has been used for successfully, just pointing out that this is a good method for trying to get at deposition, dry deposition. So also wanted to mention that another way that we get at dry deposition is we measure concentration and then we apply model deposition velocities. So that there's other ways to do this. So one of the things we use this for, and this papers were published in 2012 and 2013, was work that we did for EPRI, helping develop a total maximum daily load of mercury for Florida. And just to summarize, some of the values of using this method, we measured dry deposition rates, and then we also had used wind direction and criteria air pollutant. And using this data, we identified 3 sources of mercury-2, mobile sources, local electrical generating facilities, and transport of air from the northeastern United States. And the results pointed to different chemical forms of mercury associated with each of these surfaces. And this work also was when we really started to say that the TECRAN wasn't really measuring gaseous oxidized mercury accurately, because we were getting higher rates of deposition with the surrogate surfaces than we were with the TECRAN measured concentrations put into a model. Next slide. So the other thing that this sampler can be used for is looking at regional trends. And so the data that I'm showing you is from Mark Sather, who was retired but was with US EPA. And they were interested in looking at if mercury deposition would be reduced when large coal-fired power plants in the Four Corners region, Utah, Colorado, New Mexico, Arizona, went offline. So they did a coal-fired plant and then a post when the coal-fired plant was shut down. And you can see nicely here how just there are really regional trends in deposition going on between these different sites. And so, and we also see the same thing with work that we've done in Nevada and California. So this method can also be used to just try and get at regional trends. Next slide, please. So on this figure, I've compiled dry deposition data collected using the arrowheads across the United States. And you can see that this really is some really interesting trends. So the green are low concentrations and the red are higher concentrations. And you can see that the coastal sites on the West Coast have low deposition rates. And this is because they are primarily influenced by the marine boundary layer. Then the coast ranges, which are the 20 and 19, and these are site numbers actually for the paper that are the numbers that you see, but the values are color coded. You can see the coast range sites have higher values than the coast because they're being influenced by long range transport in the free troposphere. And then when you get to the Sierra Nevadas, we have much higher deposition. And this is because we're getting a lot of long-range transport influence, and you can kind of think about this as due to elevation. So air comes in over the Pacific, it hits the coast ranges, it gets bumped up, it hits the Sierras, and then gets lofted over the Sierras, and so you're depositing this long-range transport mercury at higher elevation. And then you can see like the mid kind of Colorado, New Mexico, these are Mark Sather sites. This is 14, 10, 11, 13, 12. You can see that the deposition is lower, and this is likely due to the fact that we're depositing the mercury that's coming in by way of long-range transport to these higher elevation sites. Nevada, where I live, has a lot of mountain ranges and is at high elevation. And then once you get into Colorado, New Mexico, the values become lower. And then, of course, in Florida, where wet deposition is 80% of deposition, you have much lower dry deposition rates. Next slide, please. So we recently have applied the arrowheads with a method that we developed that's called the reactive mercury active system. And we developed the system when we realized that the TECRAN GOM measurements were biased low, or actually we used this to show that the TECRAN measurements were biased low. And basically, this is just a picture so you can see it consists very simple pumps, filter packs. Next slide, please. And basically, this just shows the setup for the system. And we have 3 different types of membranes. We have nylon membranes, and we use nylon membranes to get at the chemistry of the gaseous oxidized mercury or reactive mercury compounds. We use cation exchange membranes to get at the concentration. And so in some settings, you can see like the orange filters in a filter pack. With that, we're just getting at reactive mercury. So that'd be gaseous oxidized mercury and particulate bound mercury. Whereas if we have a particulate membrane in front and we use the PTFE membrane from Sartorius, so if we have that in front, it gets PBM, and that results in a GOM measurement on the nylon membranes and cation exchange membranes. Next slide, please. So basically, I just wanted to show you that then what we do with the nylon membrane data is we have developed standard thermal desorption profiles, and then we have a MATLAB program for deconvoluting the profiles that allows us, so on the right, I guess it might be your left, is what a standard profile would look like in black. And then you can see that we then deconvolute the profile to get at different mercury compounds. And these include mercury oxide, mercury bromide, mercury chloride, mercury nitrate, mercury sulfate, and then an organic methyl mercury. Next slide, please. So I just wanted to show you some data from sites where we have the RMAS measuring and the arrowhead. And one of the first times we did this, we cooperated with researchers that have the Zeppelin Svalbard site. And just a couple things to point out on this slide is, one, just remember that the RMAS measures GOM and PBM, whereas the arrowhead measures gaseous oxidized mercury. So what you can see from the graph, let's see, I think it's on your right. You can see that the TECRAN calculated flux based on the TECRAN concentration is the gray line, and for dry deposition, it's much lower than the value calculated for the deposition using the arrowhead, and the arrowhead is lower than that calculated using the RMAS membranes. And the reason for the difference between the arrowhead and the RMAS is that the RMAS is measuring particulate mercury and the arrowhead is measuring gaseous oxidized mercury. Then if you look at the graph, it's kind of interesting. You can see, so what we were trying to capture here were mercury depletion events on the membranes, and we did capture mercury depletion events on the dry arrowhead membranes. But what you can see here also that's kind of interesting is towards the end of the sampling period, the RMAS is still measuring something, and it must be PBM because we're not measuring anything on the arrowhead, which is a measurement of gaseous oxidized mercury. And then on the left, I just put some thermal desorption profiles so you could kind of see the different compounds that we were measuring during these time periods. Sorry, next slide, please. So we did this also at Guadalupe Mountains National Park in Texas. And the graph, some things to point out on the graph. I put this picture in because I think it's really pretty, and this is a very unique park. It has a fish consumption advisory for mercury, but it's a carbonate reef. And so EPA was interested in whether fracking, or was oil and gas recovery is the appropriate term to use, was a source of mercury to this stream. So if you look at the graph, what you can see is the different mercury compounds in the bar graph. And then the line graph, you have the cation exchange membrane concentrations. And then you have the dry deposition measured by the arrowheads in hatched orange. And then using the RMAS concentrations and the dry deposition to the plate, the arrowheads, you can see that we can calculate deposition velocities. And I just want to point out that the deposition velocity on the 8/3/2021 is much higher than other time periods. And during this time, we had air coming in from Mexico, and we had a lot of sulfur, nitrogen, and organic compounds, and much less halogenated compounds. So just different settings. And then when we had air that had a lot of halogenated compounds, the next 3 bars, you can see the deposition velocity was lower. And then when we have more oxide compounds coming in towards the end of the project, you can see that the deposition velocities go up. So this is a way to actually get at deposition velocities that are needed for different mercury compounds using the RMAS concentrations or a dual channel system and arrowhead measured dry deposition velocity. And then this also allows us to calculate velocities for specific dominant compounds. Next slide, please. So we also did this on Amsterdam Island, which is in the middle of the Southern Ocean. It's like 3,300 miles from Africa, South America. Like, it's a long boat ride to get there, okay? And to put our samplers on this island, they have to be helicoptered in. So this is quite a big deal to get data from this location. But again, on the graph, you can see that the deposition velocities, the dry deposition is fairly constant. The dry deposition to the RM concentrations you can see in black. The dry deposition is the dashed line, and you can see it's fairly constant, but the deposition velocities vary, and that's because these deposition velocities are correlated with wind speed, highly correlated with wind speed. So, and the chemistry did not vary a lot there. We did have some unusual compounds in the beginning, and we think it's because of biological activity. Next slide, please. Yeah, so I'll just leave you with some conclusions. What's the utility? This method is a surrogate for potential dry deposition to the Earth's surfaces. It's useful for understanding regional patterns in dry deposition and quantifying deposition associated with specific sources. Trends observed at USA sites that were relatively unimpacted by anthropogenic sources show that deposition from west to east decreases due to deposition of mercury to surfaces as air is moving over the US, and also as affected by precipitation. And use of this method coupled with the RMAS or a dual channel system could allow for calculation of deposition velocities, and direct measurement of wet and dry deposition is currently the best way to understand mercury deposition to ecosystems. Next slide. I just want to acknowledge students that help in the lab, National Science Foundation funding, EPA provided some limiting funding, and we thank the staff at Guadalupe Mountains National Park for collecting and deploying the membranes. And then the Amsterdam Island mercury data was collected with funding from the French Polar Institute, and Stefan Osterwalder received funding from the Swiss National Science Foundation. So I think that's it. And I don't think I put another slide in. Yeah, okay. Thank you. I'm happy to answer questions.
Thank you, May. Do we have any questions for May?
I think everybody just wants to go have a beer.
Yeah, what's that?
Hey, May, this is David Gay. Quick question on the RMAS and the Arrowhead. Just rough numbers. How long can you leave them out? I mean, days, months, and short term too. How quick can you run them?
1 to 2 weeks to get a signal, and well above the blank. And the analysis of the cation exchange membranes is pretty quick. You just digest them and put them in a 2600, Becquerel 2600. And the nylon membranes take a little bit longer because you have to thermally desorb them.
Okay, thank you. And thank you, May. And let's thank our speakers for this session. And this concludes the scientific symposium, and I'll turn it over to Mike.